classification of cell. Based on cellular organization and the nuclear characteristics, it is divided into three types. The first one is prokaryotes, second mesokaryotes, and third eukaryotes. Most of us have heard only prokaryotes and eukaryotes, isn't it? Yes. So, let us see the second term mesokaryotes in this video. First, prokaryotes. Wherever the term pro comes, it means primitive, first or friend, and the karyon refers to the nucleus. So, prokaryotes are the first formed nucleus with no proper nuclear membrane covering. In prokaryotes, the organelles cannot be identified in the cytoplasm. We cannot find the well-matured nucleus inside the cell. These cells are mostly unicellular in nature. Example, bacteria and blue-green algae. The second is mesokaryotes. These are the organisms which are intermediate to prokaryotes and eukaryotes. So, how we can define is that we could see an organized nucleus, that is a nucleus with the membrane coverings, but we cannot identify the organelles in the cytoplasm. That is why it is mesokaryotes. Example, phytoplankton like peridium. Eukaryotes. From the term itself, we could identify that eukaryotes will have well-defined nucleus. Yes, the term eu refers to true, whereas the term karyon refers to nucleus. So, the cell with the well-defined matured nucleus can be identified in the cytoplasm. The cell also have well organized and well covered membranes of organelles. Most of the eukaryotes are multicellular in nature. The best example are human being and reptiles. There is also one more possibility of eukaryotes, that is endosymbiosis. So, what is the term endosymbiosis or endosymbiotic theory? As per the definition, when two eukaryotic organelle believed to be the descendant of the endosymbiotic prokaryote, which means when the prokaryotic cell become an eukaryotic cell, then that process is known as endosymbiosis. So, how is that possible? When prokaryotic cell grows inside in volume to become an inner foldings of cellular membrane that completely covers the nucleus as nuclear membrane along with the development of endoplasmic reticulum. When this prokaryotic cell engulfs an aerobic protobacterium which utilizes presence of oxygen in the cell, it becomes a mitochondria. And that's how the cell became eukaryotic cell, by having all the well-defined nucleus and organelle inside it. Next we move on to cell organelle. Under cell organelle we have endomembrane system, which is the membrane comprising of plasma membrane, nuclear membrane, endoplasmic reticulum, Golgi apparatus, mitochondria, lysosomes and vacuolar membrane. As we seen before, only eukaryotic cell will consist of all the well-defined cell organelle. The first organelle we are going to see is endoplasmic reticulum. It was discovered by K.R. Porter in 1948. It is found to be attached near the nuclear membrane. This endoplasmic reticulum receives information from the nucleus through messenger RNA. We could see all this RNA in the upcoming videos. The endoplasmic reticulum helps in conveying the message from cytoplasm to the nucleus and also nucleus to the cytoplasm. They are said to be the main production site. Why? Because the lipid, which we called as fat synthesization and protein synthesization, takes place here. There are two types of endoplasmic reticulum. One is rough endoplasmic reticulum and another one is smooth endoplasmic reticulum. Why it is named as rough and smooth are: in rough endoplasmic reticulum, the ribosomes are attached to the layer of the endoplasmic reticulum which seem to be rough in nature. Whereas in smooth endoplasmic reticulum, the ribosomes are absent and which looks like smooth layers of endoplasmic reticulum. In rough endoplasmic reticulum, the protein synthesization takes place. So it is the site of protein synthesis which are immature in nature and located in the vesicle. Whereas in smooth endoplasmic reticulum, the lipid or fat synthesization takes place. Endoplasmic reticulum plays an important role in transferring proteins to the cell when they are needed. The next organelle is Golgi apparatus. It was discovered by Camillo Golgi in 1898. They are also known as dictyosomes. Why? Because they are found as small vesicle in plant cell. They are referred by main processing units. Why? Because the immature protein and lipid synthesized by endoplasmic reticulum are converted into an usable form in Golgi apparatus. It stores and processes the immature proteins and lipids into a cell usable one. It plays a major role in formation of lysosomes, which is the next organelle we are going to see. Lysosomes, where lyso refers to diffusion or destruction, where some or soma refers to the cells. Lysosomes are also known as suicidal bags. These are the vessels like cell which contain enzymes performing proteolytic activity. The proteolytic refers to digestion of protein. The enzymes which destroy microorganisms that enter into the cell, and also it destroys the microbe substances if it feels that is not good for the cell. As we know, the cell is made up of full of protein. So we can have a doubt that whether these lysosomes are dangerous to the healthy cells. No. Why? Because the lysosomes are covered by an outer membrane which protects the cell from the digestion of the whole cell itself. It destroys the organelle in case if the organelle is get affected by the foreign particles or it get diseased by any other microbes. It also destroys the whole cell if it feels like the cell is infected or the cell is diseased. Some main functions of lysosomes are intercellular digestion, that is digestion of carbohydrates, proteins, lipids present in the cytoplasm. Second, autophage. Autophage is nothing but digestion of the organelle in case of diseased or infected. The third one is autolysis, which means it digests the old cell when the cell gets infected or when the cell gets diseased. The next one is aging. Aging is the disruption of intercellular molecules. The intercellular molecules which I have mentioned before are carbohydrates, proteins and lipids. Next is phagocytosis. It means the engulf of large substances which get digested through macrophages, and it also get attached to lysosome for further digestion. I will explain this in cell activity. Exocytosis, it is one of the activity of the cell which can be seen after the cell organelle. The next organelle we are going to see is that centrioles. Centrioles, which comes under the peroxisomes, which are small spherical in shape and a single membrane bounded organelle, which take part in photorespiration of the cell and glycolate metabolism. It can be found in liver and kidney of mammals in animal cell, and in plant cell it is present in the leaf cell. It has some functions. The main function of centrioles are during cell division, it helps in the spindle formation. The spindle formation and the process of cell division can be seen in cell cycle, which can be seen in the upcoming videos. Centrioles, it is located near the nucleus. It have two cylinder-like structure. Each are called as centrosomes. Centrioles present only in the moving plant and animal cells. Why? Because the cell division that takes place in plant is different from that of animal cells and moving plant cells. In plant cell, Golgi apparatus plays an important role in cell division. The spindle formation cannot happen in plant cell. That's why the centrioles are absent in plant cells. Next is mitochondria. It was first observed by A. Colliker in 1880. Altman, who named it as bioplast. Later Benda, who named it as mitochondria. It is a double layer membrane present in the cytoplasm. The number of mitochondria depends on the type of muscle the person have. For example, red muscle fiber has more number of mitochondria. The sports people or the person who are working more or giving more stress to their muscles, then the mitochondria increase in their number. The muscle getting strengthened during physical activity, which ultimately leads to the stamina, as we all know. So this is all because of mitochondria. If your stamina increases, it means the number of mitochondria in your muscles are increased. Why? Because mitochondria is also known as powerhouse of the cell, where in mitochondria the energy is released by the process of converting ATP, that is adenosine triphosphate, into ADP, that is adenosine diphosphate. The adenosine triphosphate gets converted into ADP in the presence of oxygen and it releases energy. So mitochondria, we already said there is a double layer membrane. The inner membrane contains many foldings. These foldings helps during the conversions of ATP to ADP by expanding and providing space for the conversion. That's how it saves the energy, and it is also named as powerhouse of the cell. That's all about the cell organelle. Now we are going to see the cell activities that takes place in the cell organelle. There are two types of activity. One is endocytosis and another one is exocytosis. In endocytosis, the function is grasping or engulfing of an substance into the cell. If the plasma membrane feels the substance is good for the cell, as we seen before. So what it do is that it engulfs the substance through the cell membrane. The cell membrane gets surrounded to the substances and it completely engulf and form a vesicle and drag it to the center of the cytoplasm. This is what happens in endocytosis. So there are two subtypes in endocytosis. One is phagocytosis and another one is pinocytosis. Phagocytosis is that it engulfs the substance, what I have said before. Pinocytosis is the fluid droplets. So it engulfs the fluid droplets as the process I have explained in phagocytosis. Same process happens in pinocytosis, but in phagocytosis the substance is engulfed. In pinocytosis the fluid droplets is engulfed. Now we can relate the macrophages which we are explained in the function of lysosomes with this phagocytosis process. So the macrophages, what it do is it engulf in larger substances and it gets digested by off and it gets connected with the lysosomes for further digestion. The second process is exocytosis, where the substances or the particles are pushing or making the substance to move out of the cell. That is what exocytosis. The pushing or making the substance to move out of the cell is also the same process which we are done in the endocytosis.